It's my pleasure to introduce Professor Fiona Brinkman. She's with the Department of Molecular Biology and Biochemistry here at SFU. Fiona has many awards and accolades, the most recent of which is appointment to the Board of Directors for Genome Canada. So congratulations on that one. That's fairly recent. And Fiona's going to be talking to us about genomics, bioinformatics, and HPC. So Fiona, go ahead.
Thank you. No, it's my pleasure being here. I have to say, being asked to talk about how essential WestGrid is to my research is like saying how essential food is to my survival. So it's really a no-brainer. But what I hope to do is just sort of very briefly illustrate, what we're trying to do and how we're doing it, and how WestGrid has become essential for that. So first, you have to understand what I'm interested in is infectious diseases and improving approaches for infectious disease control. And this has become a real problem, infectious disease control, as illustrated in this first slide. Globalization in particular has caused very increased risk of spread of pathogens, as shown in this classic well-traveled salad. Most of your food alone is coming from different locations, and it's very easy for a pathogen to spread rapidly thanks to international travel and food being disseminated more widely than it was even a decade ago. So an example is this German E. coli outbreak you might have heard of a couple years back, where literally 20 countries were affected within a month. And these are significant impacts. Not only were there over 50 people dying and over 4,000 becoming ill and associated economic losses for that, but the farmers claimed, €417 in losses. They were compensated 220 million euro by the EU. So these have serious impacts, and we need to come up with faster, better responses for basically infectious disease outbreaks and general infectious disease control. And then add to that, we have this problem of antibiotic resistance, which is really increasing. And basically the problem is you have these infectious disease bacteria, as sort of illustrated by, let me see if I have a mouse here, as illustrated by the purple bacteria here. And then you have, let's say, good bacteria. You actually have lots of bacteria in your body. You're actually full of more bacterial cells than you have human cells. And you have lots of good bacteria that play important roles. But when you take an antibiotic, what happens is you're killing off those infectious bacteria as well as good bacteria. And any resistant microbe, in particular pathogen, can just multiply with no competition. And so there's a real problem with us selecting for these resistant microbes. And in fact, in this graph shown below, basically any antibiotic that we've developed within about a decade, we're seeing or less starting to see resistance occurring to that drug. So we really have to come up with new approaches, or we're going to really be at high risk of having serious impacts worldwide. So my interest is in developing more sustainable, what I like to call holistic approaches for infectious disease control, looking at the pathogen, the host, and the environment that all play a role in developing disease. And I'm just going to highlight a couple of brief projects, but just so you generally know, what I'm interested in is things like targeting pathogen virulence, basically identifying anti-infective drugs that disarm a pathogen rather than killing it. So that pathogen is actually still around. It's not, there's not the same selection for it to develop resistance. It's just not able to cause the disease. And also, I won't get into this at all, we can develop immune modulators that dampen down the damaging inflammation. Most of the disease that occurs, infectious disease, is actually your own body going wonky with your immune response and actually damaging yourself with this strong inflammatory response. And if we can dampen that down and boost a good immune response, that can also be another approach. And then lastly, an area we're getting into more is really looking at the environment, integrating social network data and other kinds of data to better ID the source and cause of disease outbreaks. So, and I'll just add a note that we've been using these methods, we've developed also for other areas like environmental toxicology, studying allergy and asthma, et cetera. So, but what I'm going to do now is just talk about two areas where the first one about anti-infectives is disarming the pathogen I'm going to mention at the end in one slide. And I'll illustrate this, talk first about this examining the environment approach, because it sort of illustrates some of the needs we have and the different kinds of needs we have. of which probably one of our biggest is we have increasingly this problem of sort of requiring all against all or many against all comparisons of the DNA sequence data that we're analyzing. Usually when you have infectious disease, you're wanting to investigate drug targets, finding genes that are specific, or you're trying to study a new pathogen and understand more about it, or you're trying to look at the microbiome, as I'll talk about, or all the microbes in an environment and understand which are pathogens, etc. And all of these analyses are really complicated by the fact that as we sequence more and more bacteria, we're getting a lot more data. And as illustrated in this nice slide from Lincoln Stein a few years back, sorry, I'm having trouble with my mouse. There it is. You know, this is your classic hard disk storage megabase per dollar, doubling time, you know, a bit over a year. And then you've got your, before we had sort of as rapid sequencing, or what we called next-gen sequencing or NGS sequencing, we had a doubling time about 19 months for the cost to sequence a base pair of DNA. But now with the new technologies we have, this doubling time is now 5 months. And in short, well, of course, we never know if it's going to continue like that. We definitely have a problem where it's almost becoming more cost effective to just resequence than actually store the data. But there's a lot of challenges regarding that, and a sort of who has access to the ability to resequence versus not, a lot of ethical issues. But in short, we're facing this big deluge of data and needing to come up with better ways to handle it. And basically at this point, increased compute power and storage are key in order for us to basically handle all of this data. So this is just one little example of something that we've developed where we do have to do this all against all as this Ortholuge DB. It's a database of where we just do sort of all against all of all the bacterial genomes, about 2,000 or so. And we're identifying all the genes unique or shared between different strains, for example, finding genes that are found only in pathogens and not in non-pathogens, or unique to certain strains or species that you want to identify with for suitable drug targets or diagnostics. And just to give you a sense of timing, you know, just to run an update of one component of this analysis where there's about 16 trillion gene comparisons with about 200, 400 cores on the Bugaboo cluster. That took about a month to run, but the problem is this is going exponentially, so we certainly have huge challenges ahead, not just in compute power requirements, but also needing to come up with more efficient methods. But just getting to, I just want to profile this environmental associated project, because it really illustrates that it's not just about analyzing the data we have, but also other challenges we have, like coming up with assessing accuracy and simulating some of our data in order to develop better methods. So this one project that I'll highlight is a Genome Canada funded project that's using what we call metagenomics to transform water quality monitoring, basically using what we call Watershed Metagenomics. And so to define what I mean by that, so metagenomics is a study of all genomic material recovered from an environment. And essentially, by sequencing literally all the DNA in that environment, it helps describe all the microbes in that environment. You can identify those, what microbes those sequences may belong to, and also what they potentially can do based on what their gene sequences encode. And then a watershed is basically an area that drains all precipitation from an area into a river or set of rivers. And the idea is that water is affected, water quality is affected by everything it comes in touch with as it drains in. And some watersheds are very nice and clean, like our in Vancouver here, greater Vancouver area. You know, in Vancouver we have these watersheds, the Capilano, Seymour, and Coquitlam watershed that serve water to different parts of the city, and they're, you know, really nice, clean water that are protected. But of course, there's some watersheds that are not so good and can cause real problems. We have significant issues with some populations that have 365 days a year boil water advisories because of fecal feces contamination in their water. of an unknown source. And we have, in general, a lot of expenditure to track water. That's very important because you could have outbreaks of illness due to bad water that can cause, again, significant health and economic impacts. So Watershed Metagenomics, basically what we're doing is trying to understand all the microbes in these watersheds. And the reason we're doing this is because the current emphasis of water quality testing is at the tap, when we really should be looking more at the source. We should be trying to find where are these pathogens coming from and try to stop them at the source. So for example, if a farm is has waste from animals that's going into a river, we should be stopping that at the source rather than just trying to treat the water as it comes down that river so that we can drink it or others can use it. But the other issue is the fecal coliform test is very inaccurate. And this is a standard test. You might hear coliform counts for closing beaches. But the problem is it doesn't detect all pathogens. It only detects those that are coliforms, this term we use very generally, and not all coliforms are pathogens, so it actually can give you a high coliform count potentially, but not necessarily be a problematic water. So we need to come up with better water quality tests to detect fecal pollution. We need better indicators of bacterial, viral, and different kinds of microbes, and essentially environmental survey modeled over space and time is needed to find these. And metagenomics really is a key tool to do that, because what we can do is sequence all the DNA in a control watershed in samples over time, a human impacted watershed, an agriculture watershed, and then compare those and those sequences that we get from those different watersheds over time and space, looking upstream and downstream of, say, a pollution site. We can then do modeling and biomarker identification, identify new markers that we could use, DNA-based markers for water pollution. And another nice thing about that is you could potentially link the source. You know, if you can find a marker and you get that sequence, and then you can link that sequence back to this particular outhouse or whatever, septic tank or whatever, you can potentially clean up and know for sure that that is the source. So the challenges we have are a few. One challenge we've really faced that is different from just analyzing your data is that there's many new analysis methods to be evaluated that are always coming out. This is just a sample. And really what we need to do is simulations of data to evaluate these analysis methods in controlled conditions, and they take time. So this just sort of illustrates you know, how speed can be an important consideration when looking at these different methods. This is I've got some bits of terminology there for anybody who is more familiar with analyzing a metagenomic sequence like this, but for those who don't worry about it. But in short, there's these sequences that are being analyzed on Bugaboo, and a combined total of 140 trillion comparisons. And in this case, we just did one little comparison on one node, one core, and then we're just trying to see, you know, how they perform. And as you can see, they're really different. And methods like that are more using this one approach called BlastX are significantly more computationally costly than other methods that use RAP search to this other method. And I won't get into this, but just to say that these the accuracy of these methods really varies notably, and so you know, but it's not at the level. So this is like a method called Discriminate, which is one of the better performing methods for RAP search versus BlastX, and the difference in accuracy isn't that significant versus the computational cost and time. And if I won't get into this, I can always ask. You can always ask questions later. But in short, the point is that we have to do these simulations of data to assess accuracy, and that alone can take quite a lot of time to do. So but then the analysis of water samples themselves, you know, there's quite significant data. And one of the challenges we face is often needing short bursts of runtime. we ideally want to sort of run something for a bit, look at the data, and then run it again. We don't have the big long run times or long consistent need for cores and other fields like in physics perhaps. But just to give you a sense of amount of data that is embarrassingly parallel workflow, essentially, we're looking at 7,710 days to date for what we've had to run just in the last while. But what's notable is this is actually leading to us IDing potential new markers of fecal pollution. And there's no way we could have done this without WestGrid. Absolutely. This was essential. And so what we're able to do is basically within this 3-year length of this project, we're aiming to identify these new markers of fecal water pollution. And then we're just starting, we still have more analysis to do, but of the analysis we've done so far, preliminary analysis, we've got some sort of preliminary markers. And the nice thing is we have this, we're working with the BC Centre for Disease Control, who actually centralizes water quality testing in BC versus other provinces that tend to have it be centralized through Environment Canada. And they're actually going to be implementing PCR-based tests and actually directly use this to start monitoring water quality with more markers. So the other thing I wanted to bring up was this issue of pathogen virulence and coming up with approaches where we're IDing anti-infectives. I'm going to do this pretty briefly. Actually, I don't have my time. What's my time? Okay. Just very briefly, I just want to mention that we have other kinds of needs as well. Those kinds of needs are very sort of sequence comparison against sequence comparison. We have these approaches that are more doing three-dimensional docking of compounds where you're sort of literally trying to identify small molecules that can bind to a particular protein in bacteria. And in particular, what we're interested in, we have a few different projects, one involving industry collaborator that I shouldn't really talk about because of IP, but we have other work involving where we're interested in toxins, and basically what we want to do is come up with compounds that can bind to these toxins and basically inactivate them and allow the bacteria still to live. So these are compounds that are not toxic to the bacteria, but bind the toxins and inactivate them, and so stop them from causing disease. And so what we do is we take these compound libraries of, like, for example, the zinc library of 11 million compounds, or we have smaller libraries of known drugs where we're trying to repurpose drugs for different potential uses. And then we're looking at those in all these different conformations and seeing how these drugs interact in a pocket in a protein. And just to give you a sense of time, roughly running this glide software using 100 bugaboo-like cores, for lack of a better term, takes about 12 days runtime to identify possible drug leads against just one possible protein. We have had some licensing challenges with that, but in short, we have been able to find already an existing drug that's currently used to treat osteoporosis that may be repurposed as an anti-infective that just happens to have this activity, anti-infective activity as well. And so these kind of analyses, again, is where HPC is absolutely critical, because the amount of time to look at all these possible protein targets in an organism, that has thousands of different proteins, and we have literally hundreds of targets that we're potentially interested in, definitely takes time. Okay, so it really sort of ending here now with a new direction we're going in now, this IRIDA project, which stands for Integrated Rapid Infectious Disease Analysis. And what we're interested in doing is really taking a lot of these different ideas for how to better control infectious diseases and using genomic DNA and DNA data, and basically trying to integrate it together into a platform where when there is signs of an outbreak occurring, we can very rapidly sequence the genomes of those pathogens and use those by overlaying other kinds of data and epidemiological data, social network data, other kinds of data about the pathogens, and environmental data, we can start to really learn more quickly, how this thing is spreading, where it's potentially coming from faster, so we can implement a more rapid response and literally, hopefully, come up with a situation where we're avoiding, the sort of international spread, which is always the sort of worry with a lot of these, when these early outbreaks start. This is a collaboration with the Public Health Agency of Canada National Microbiology Lab, Gary von Domsler, and is also associated with the University of Manitoba, and Will Sow is at the BC Centre for Disease Control and the Public Health, BC Public Health Reference Lab, but also involving a bunch of collaborators around the world, and I'm really enthusiastic about helping to make this happen. So in closing, we are aiming for more sustainable infectious disease control, really wanting to have more accurate and rapid pathogen detection, identifying the source of infections more quickly and more accurately, and using anti-infective drugs, or trying to use evolution to our advantage to select for basically converting pathogens to become non-pathogens, rather than this concept of always trying to come up with new drugs that are killing them that they develop resistance to. This R&D basically does have notable HPC needs, both storage and processor requirements. Haven't really gotten into the storage, but we have a 60 terabyte allocation, which has been really useful. And then WestGrid basically has been key, not just for this work, but I'm really seeing a growing body of projects in biology and medicine. I'll be interested to see how many WestGrid applications there are in this area for this next round of allocation assignments, because I'm seeing a lot more people sort of who are not traditionally very computational, who are really needing to use WestGrid to perform certain analyses. And these really are translating into developing, identifying new potential drugs, pathogen detection tests. I mean, what's really nice, gratifying, is we're able to translate this to really practical things now. And I'll just add two comments. One is just having this WestGrid allocation plus the capability to handle short-lived intense jobs is really useful. Like, we were able to get up to 400 cores for a few days and then take a few days to analyze and then do it again. That kind of capability was really appreciated on a short term when we really needed it. And also, just to say, I really welcome, as somebody who's been applying for these allocations each year, and when you have a 3-year project and things aren't really changing in terms of your allocation needs, it's nice to have that more streamlined approach for reapplication. So thank you for setting that up. And I appreciate there's lots of growing pains as we sort of adapt, things, but I think it's really helpful. And, just really want to emphasize that WestGrid has been a key base for enabling all this to happen. I want to acknowledge a bunch of people who have been key for this. In particular, a shout out to Matthew Laird, who's in this room, who has been really maintaining all of our computing resources and dealing with various issues that come up, and really appreciate his work. And also, I'd like to acknowledge our great team that have been doing the drug discovery, and I have to do a special shout out to Erin, who's in the room. Happy birthday! Just thought I'd publicly announce it across Western Canada. So Erin is doing, and check out this cake, Erin. It's got like little protein alpha helices on it. It's awesome. So she does a lot of this docking. And last but definitely not least, I have to just say this absolutely key support. And I was kicking myself. I realized I should have the ERMACs on there, because ERMACs have been so critical for us here at SFU. But Martin Lixon and Atta, for just having that great WestGrid support is really key. And with that, I think I'm done, and I'll be happy to take questions. There's time.
I think we have, we got a fair bit of time. I mean, the goal for these sessions are to have lots of discussion afterwards. That's why we try to keep the talk kind of short. So what we'll do is we'll just open up the table. I see I have a question at UBC, so UBC can go ahead.
Okay, start. Hi, my name's Twee. I had a question about, so you mentioned you have really long run times, hundreds worth of days or so, I guess core times really. I'm wondering how much of your time is actually spent waiting for your jobs to run. So how much of your time is actually spent in the queue versus actually running?
That's actually a good question. We were having an issue with that recently, weren't we? Thea, is she here? Or do you want to put over at Mike? That's the advantage of having my lab members here. But it is a good point that, when we're calculating read time, we were actually discussing that when I was getting some stats, what the latest stats are, that you do have that issue of, just waiting in the queue versus actually running. But yeah.
Yeah, I think it's pretty variable in terms of...
Put the mic, or is the mic close?
It's okay.
Okay.
Yeah, it's pretty variable. I think sometimes it runs right away, and then sometimes maybe you wait about half an hour in severe cases when we've been using a lot of our allocation, for example. Then it can be a lot longer, a day or so. But for the most part, it's really quick, like within half an hour.
And for the jobs that actually get to run, how long do they run for?
So that's really dependent on the kind of jobs. So I have some that run for like 5 minutes, and then some I think the longest jobs that I run are about 24 hours.
24 hours? Okay, cool, thanks.
I've gone up to like 100 hours.
And I'm also in the group, and I've gone up to about a month.
Yeah, it varies depending on the particular analysis. And one thing that's nice is this embarrassingly parallel issue is, it's not like we can sort of technically split things up, right, if we have to.
And
yeah.
By splitting up like that, what I've been told by our local team here, one of the fantastic things about having embarrassingly parallel, being able to split up, is you can fit in those little holes in the scheduler, because we're not running these 2-week jobs that you typically would see in, say, physics or chemistry. We say, we only need it for 6 hours. There's these little slots that open up in the scheduler. We can slip in there and get that job running. I think I'm explaining that right.
Are there any other questions out there? Feel free to just wave your hands at the room you're in, and people will turn on mics for you and things.
Oh, there's somebody waving in the far left.
Go ahead.
Doug, I think that's you. Go ahead.
Go ahead from Calgary. Yes, I'm Doug Phillips. I'm one of the support people for WestGrid here. I was wondering about training issues. WestGrid is not really a good platform for learning new software. It's intended more for production computing. So I was wondering if you can comment how your research group is getting the training in all these new software products you mentioned.
Oh, so I'm going to ask these guys. I'm actually really curious to have you guys comment. This is a fantastic opportunity, I think, to get some feedback from people quickly, rather than, sending out a survey and then not getting responses. So we can corner them and get them to give answers. But I will just say that, I think we've been tending to use a bit of a model of, that there's somebody in the lab like Matt or something who can sort of point people to things. And certainly, I think there is a real challenge with certain types of software that, we tend to feel that, certain things are easier to do on WestGrid than others. And, like we have this license issue with one software where it requires it's by IP and MAC address, and we need to somehow get it to see the cluster without it seeing outside, and so there's certainly different kinds of challenges. But I would love to hear if you guys have comments on the training.
First of all, we have our own small cluster we can do prototyping on, and luckily with the very short queuing times we've seen, it's easy to try things out more or less. Is that?
Yeah, that's right. So I guess our model definitely is trying things out on our local servers, and then once we see that it's working, then we can run it on Bugaboo. One thing that's been great too is the WestGrid support for helping us to install libraries sometimes when things aren't working. So that's been really responsive in terms of helping us set up programs on WestGrid.
Maybe we shouldn't be saying publicly how easy it is to get things running on Bugaboo. We want to keep it nice and quiet.
Yeah, but do you have any thoughts, the one just like when you first came here, what it was like to get going, or what you found, what you needed, or resources, or?
Yeah, there was definitely a learning period for sure, and I guess there wasn't a really structural learning environment. It was sort of I ran stuff and it didn't work, and then I ran it again and it still didn't work, and eventually I figured out how to make it work. But so there wasn't a structured learning program for myself. I don't know what your.
Yeah, it was trial and error.
Like any good research.
Yeah. Yeah.
I think especially with, like Mike uses a lot of different tools, and you saw that big list, he has to try to use them all. And so a big part of time is trying to set up these tools and actually get them running. And that is something you just get good at with experience.
And I guess I would put to you, if there's any ideas you had about training you want to bounce off us, and certainly I think these are good sort of people that could give feedback, I guess, on what would be reasonable.
So I think we have one other question at UBC.
Hey, so actually, Fiona, we probably know each other. I'm Niels Hansen. I'm at Stephen Hallam's lab. So I have a few comments about, I guess, the algorithms and sort of the things that we've been trying in order to get, performance, because, well, we missed the boat on applying for a WestGrid allocation last year, and so we were, restricted on how much we could use. And so we have a number of ways that we sort of got around it. So we have our own, we hooked all our, we have our own grid in our lab of about 30 Mac Pros, which we have now a SunGrid engine running on them. And so again, we have a similar sort of test environment where we test out our software. But I'd actually be really interested to see, because you're doing quite an expansive test of all the binning algorithms that I'm really curious about. But what we noticed is that there's a sort of an improvement to the BLAST algorithm called LAST that's out of Japan, and in practical terms, it's probably about 100 times faster, such that we can run like our an Illumina metagenomic sample, some of our larger Illumina metagenomic samples even locally. In about 2 hours per sample. So like scaling this up to the grid will probably have a pretty significant improvement. Just it's not even multi-threaded. So I mean, we're working on MPIing that code and making it multi-threaded, and then also coming up with like we're working with the DOE at the Pacific Northwest National Labs to do a Phi last for optimizing it on Intel Phi cards, which are the newest and greatest things that are on all the fastest supercomputers. So first there's LAST, and it sort of gains its speed out of when it does the seeding and extending. It has a more efficient.
Sorry, I just have to interrupt you, but I just wasn't hearing what you're saying. Are you talking BLAT? Or what do you, I just have to
called LAST.
Oh, LAST.
LAST.
Okay.
And so
yeah.
we really like this thing. And in our tests, we see it agreeing up to 98% with the BLAST. So, I mean, it's sort of a no-brainer.
Absolutely.
And,
absolutely. I mean, yeah. Or go ahead.
And the next thing is, so this all-to-all comparison is really a problem of the fact that there's no data structure on sequences to sort of have a directionality. And so you can't build a tree. So you can't sort of like isolate kind of the proximity of where you think your hit is going to be. So the consequence is you have to continuously do this like all versus all for everything. So we're interested in doing a database clustering of sequences such that you can perhaps, now this is more open-ended, and we're not sure if this is going to work or not, but that's what we're thinking of trying, is having a low, like a sort of an HMM model up here at the low clustering, so that you can sort of dictate like which general section of the database you're going to go, and then go all the way down, and maybe not even do a BLAST, because BLAST is error-prone, and do a fold-on Smith-Waterman down at the bottom, because you only have 100 sequences in this cluster. So that's another framework. And then about the
before you go too far, because I'll forget the first things, I'll just say I think this is great you're bringing this up. This is exactly the kind of problems that, I mean, BLAST is quite frankly just awful. It's this huge memory hog. It's really inefficient, and And, as we saw, like with RAP Search 2 is another example of something, another approach, just definitely considerably faster. And I will just mention that there are definitely people working on these issues of trying to come up with more efficient all against all and updating your all against all analyses more efficiently, et cetera. And I can definitely email you and point you to some people I know that are sort of working on that. Unfortunately, I think there's been a tendency to sort of take the lazy approach of just, well, let's just go, because we can. But certainly, I think there's a huge need to develop that further, and that it does give me some hope.
And also, people are very comfortable with what they know, and especially if they have a long-term project, it's like what they started with in the front. They don't want to jeopardize the results by changing the process 3 years in, right? And so sort of you're kind of locked into what you decided to do methodologically at the very beginning.
Yeah. But definitely I think people are realizing that BLAST is not the way to go for some things now. Yeah. Yeah.
Finally, my last comment is sort of to deal with the scheduling issue of always waiting in the queue. So in Compute Canada, you're going to be given a large an allocation that's very distributed over a large number of grids. Now, each of these grids the problem with this is that each of these grids is quite homogeneous, and so you need to be able to dish out your software to each one and then run it. And so you sort of have what this is kind of a master-slave problem, where you have a bunch of workers that may or may not get your task done in the time that you want. So you sort of want to dictate your own sort of master, and I'm like, okay, I'll give you jobs, and you got that done really quick, so I'm going to give you more jobs. And sort of we're thinking about a very homogeneous distributed system for metagenomic analysis, which we think is sort of the if you say you don't get a WestGrid allocation, but you're given the standard allocation, so you can still take advantage of this distributed system. And so we're working on an area, sort of a submission engine to sort of control all the grids. And I mean, we're trying to work with all the different grid submission. The other problem is not only the grids are there, but the submission is also very homogeneous. So I mean, they're similar but different, and so you have to work out all these peculiarities. The devil's in the details, but we're sort of seeing very good performance and from this kind of system, and so that's how we're dealing with the waiting problem.
Sounds great.
So,
yeah, I'm really,
yeah, we can take it offline and compare notes sometime.
Yeah, definitely.
Maybe get Stephen involved with the conversation.
Yeah, that'd be great. Excellent. Oh, do you want to say something?
Niels, it's Matt here. Yeah, we should talk because I have written a similar library to do that, but it's only focused at Bugaboo, and I'm not sure what WestGrid support thinks of automated SSHing in and submitting things. Okay, I'm getting a shrug here. Yeah, I've heard that similar, so it is very handy to have that.
I mean, it's what you would do by hand. Like if you're trying to farm the grids, I mean, to get cycles wherever you can. It's just I've got now a software that sort of is my system of how to do that.
We'll chat later.
Okay. So we have a couple more questions. So lots of questions. So great discussion. Calgary, do you want to go ahead?
Yeah, okay, it's Doug again. I was wondering if you can comment on the relationship between Genome Canada and Compute Canada as they might evolve, whether a lot of the computing will move to the Genome Canada sequencing centers as opposed to being on something like WestGrid, or what are the different roles there?
Yeah, so I could just say that definitely Genome Canada is very keen on Compute Canada. They had somebody come in and talk recently, as part of a meeting. That basically, I think there's a lot of enthusiasm for using Compute Canada resources, but there's a lot of questions right now, and there's some evaluation going on about what should be done and what are really the best way forward. And there's some sort of reaching out through some of these I think, and different initiatives for trying to come up with better approaches for just standardizing some of the standard sequence analysis methods that I think are going to be key. And in short, I see Compute Canada playing a major role still, but I do think we need to have a bit more, communication about what that role is best should be. That involves also sort of wider consultation. Make sure there's a lot of once we have sort of some ideas of approaches, that we really make sure that everybody's consulted across Canada first before finally going with it. I don't know if that really answers your question enough, but it really is. It's not a case of that I see Genome Canada moving in the direction of wanting everything to be at these centers. So they do treat these science and technology innovation centers or STICs very seriously. They're a very important component, particularly as technology is changing so rapidly for genome sequencing, and these centers are absolutely key for keeping Canada competitive.
And I think we have another question at USask. Want to go ahead in Saskatchewan?
Yeah, it's Joanne Dillon from USask. I'm just asking about this. Hi. I'm just asking about the small molecule protein interaction. Is there a sort of a electronic library of small molecule structures? How did you accomplish that?
Yeah, so there's this big zinc library that you can get that is sort of just a bunch of small molecules, and then you can get other libraries. And Aaron could maybe comment more, but basically, first I just have to say this is my PhD supervisor, fantastic person, great mentor. So I'm just so happy to see you, and I'll just almost get a tear in my eye. But I have to say, I have to say it was really there's a lot of different libraries out there. I think one of the challenges becomes that you can find an interaction with a molecule, but can you actually get that molecule? Can you actually purchase them? And some of those challenges. For one of our projects, we focused on just this little library that you can get easily of just known compounds. You have things like drug bank, I guess, is one, right? You would say, but there's others that have known drugs that are already approved for use in humans, and obviously that's a nice low-hanging fruit if you can get something to work that's already known to have the safety efficacy studies. And then what we're doing is trying to do make derived versions of those compounds that are subtly different, but still have the same activity. As one approach to finding compounds. But in short, you can get these no problem. And I can definitely pull you to that.
All right, are there any other questions out there? I don't see any notes about anybody sending any messages, so I... UBC again? Go ahead.
Okay. Hi, it's Tui again. So I used to work in a lab with a lot of biologists, and I found that trying to get them to use WestGrid was extremely difficult. And part of the reason was there's a steep learning curve, and in addition to that, the cost or the effort-benefit ratio isn't that great. So I'm wondering if you only use WestGrid for highly automatable, highly repeatable items, or if you ever use it for anything ad hoc. Because we found that setting things up, you find a cluster and it's great, and everything works there, but the queue time is too long, so you have to set it up on another cluster, and you find that everything's not set up. So you lose a week or two every time. You have to change clusters, and in the end, maybe the runtime is only 2 weeks, but it took you 3 weeks to set it up on WestGrid. Sorry, I'll just go back to my question again. I'm curious, at what point do you guys decide to use WestGrid, and at what point do you decide it's too much effort, I'm just going to use a local cluster?
Yeah, I have to admit that just us having a local cluster does increase that energy, activation energy to moving over to WestGrid, because it's easy for just some little thing to sort of run it locally, and we do tend to use WestGrid for sort of larger items where it's something that we think we can is very easy to run through WestGrid. But I have to say, I'm just, even in the past year, I'm just, as somebody who keeps getting, contacted by different researchers with bioinformatics needs, I'm definitely seeing an increase in people who are contacting us just wanting to know how to, can you give me advice on putting in a WestGrid rack allocation, rack application. So it's definitely changing a bit. That I think more people are willing to put in that effort. But I do have to say I agree that I think there is that bit of activation energy of just getting to make it worth it to run something, and it would be nice to change that. That said, I was just on a teleconference 2 days ago where somebody was saying they were really using WestGrid a lot, and honestly that was a call from across Canada, so I don't even remember where they're from, but sorry, apologies, I could find out though, but basically I definitely seen people, there was a conversation about, for this project just starting up, how much to be using WestGrid versus other resources. And in short, there was some definite positive feeling that WestGrid, that you really could count on WestGrid for doing a lot. And so I think it is changing, but I don't know if you guys have any comments about that. Would you agree that in general it can be, if you have things set up, locally and stuff, and you get something working, you're only going to move it to WestGrid. But honestly, I have to say that is, I think, really one of the best intentions for WestGrid at this time, is just for those big HPC needs. Now, I guess I would ask you though, are you saying that you would like to see more of people using them for smaller needs?
For my other lab, this is the Bespoke Lab. We did a lot of genome assembly, and we didn't have a set pipeline. We were working things out, so we didn't have scripts that would do everything. So a lot of our time was just spent trial and error. Does this work? And we tried to use WestGrid as much as possible because our local clusters didn't have the same computing power, obviously. But we found that in the end, we actually gave up after a while because the setup time was too much, and we would find our jobs would fail because we would hit the wall time of 3 days, and then we would wait in a queue for a week, and then hit a wall time of 3 days, and it's things like that. There was a lot of effort trying to get this automation, but we weren't at that stage yet. It may be later on, WestGrid would have been really good for us, but at that point, we needed WestGrid's power, but we weren't able to manage. We weren't able to manage WestGrid's queuing and resource limits with the personnel resources that we had.
Can I just comment a little bit about that? So can I? I work here here at UVic with a lot of researchers. So so the problem sometimes the researchers, they come to us and are asking, they are bringing a solution to the problem instead of asking instead of bringing the problem, they bring a solution, and that's actually causing us. It can actually sometimes cause problems to both the computational resources and to the researcher. So basically, the problem I have with the new researchers is that for example, they start running one job at a time, and they have this habit of running things interactively. So they cannot wait for the job to be in the queue and get, so they are not used to this batch mode system that most of our clusters are using. But actually, once you get to, once I take them step by step and they are used to that mentality of having, sending a job, go and do something else, sending a bunch of them, having something running, checking those that are running, checking the logs, looking at them, killing that job, sending another one. They actually get to really enjoy it. So it's a problem. It's more like the problem of the user side. So if you ask anybody, for example, if you ask Martin, oh, I have this here, I'd like just to do a quick testing, he will tell you, well, can you please submit an interactive job? So you just submit this Q sub minus I, you get into there, you quickly see what's going on, then you submit the big job to there. So I think it's more like to the user and to the end user that he should look at ways or change the way he uses these systems.
Yeah, I agree. I think WestGrid is really great for, I suppose, metagenomics, where we're doing a lot of small alignments, where you don't need a lot of memory, just a lot of processing power to get your parallelizable jobs going in a massive parallel batch. But for different types of problems, genome assembly, where maybe you need a terabyte of RAM, it's really hard. It's really hard to get a pipeline for that going that will fit WestGrid well.
Yeah, that's right. So there is this, like when you have some exception in the workflow, then I think the best is to contact WestGrid, our support at WestGrid SA, and we'll be able to, to address this. Because actually, so we are trying to offer the best quality of service out there. So, and if we don't hear this kind of, issues and things like that, we will not be able to improve. So that's why I think this should be taken offline, and Martin, I believe, or Roman at UBC, he will be able to really one-to-one, and because this is, I think, an important comment that we should address really seriously.
So I think Matt had a comment. Again, just comment to the other researchers out there. What we found is the local support team is the first people to reach out to, and they've been fantastic in helping us getting these things running when we have that problem of why doesn't this run, how do we optimize it. But so I would say if anyone is in that situation listening, to reach out to your local support team and work with them on getting the software running. They have always helped us very quickly and gotten things working in a very timely manner so that we can do these larger jobs that we need to do.
Yeah.
And in addition to that, I would be more than happy. So if you just get my contact, just give me a call. We will try to get you set up and running nicely. So we'll do our best. So we have SharkNet, we have SciNet, we have all this kind of stuff that we can get you on.
But it is a good point that it might be helpful to, when somebody new comes on, to really emphasize that they should contact the local support about getting help about running things, because they might tend to think that they just sort of get this allocation and they have to use it. Because let's face it, most computing environments, other kinds of computing environments don't have support. They don't, very much. So I'm thinking of, like Amazon or whatever. But, so it would just be good to really, I think, emphasize that for new WestGrid users.
Yeah, and I also think that people are reluctant to send email to support at WestGrid, and I would encourage them to do that. If you have problems and issues, there's a fairly substantial team that can help out with those kinds of problems. So don't be shy. That's what I always say to people. Don't be shy to ask questions. I had one comment that I needed to add. Lindsay, the executive director for WestGrid, wanted to add something about the Genome Sciences Center. They're working. WestGrid's working on an agreement with the Genome Sciences Center to have them as a kind of a node within WestGrid in some way, shape, or form. That's in the early stages, but she wanted me to mention that if that comes to fruition and that works out, that will probably help out with some of the life sciences support that WestGrid provides, because we'll have, I think, probably better workflows and things like that. Because of
a lot of things will be installed already, and that'd be great.
Yeah. So that's in the works. So let's thank Fiona for a great talk. So.